Hi everybody, welcome back. Good afternoon. So here we go. As I told you, we'll be starting from the differences between the different microbes on a cellular level. So right now, this is what we're going to talk about. See, what you need to understand is this is where we'll be differentiating cells of different microbes. Okay, we're going to differentiate cells of different microbes. So if you're talking about the cells in this topic, I believe everybody should understand that viruses would be out of this discussion since they don't have a cell. Remember, viruses are acellular, so these guys, they will not be in this discussion as they're acellular. But if you talk about the others, for example, bacteria are prokaryotes. Bacteria are considered to be prokaryotic organisms, and fungi and parasites, we can call them as eukaryotes. Bacteria are prokaryotes, fungi and the parasites, they are called as the eukaryotes. Okay. Now, technically, what you all got to understand here is that humans are also eukaryotes. Humans are also eukaryotes. Since fungi parasites are eukaryotes, humans are also eukaryotes. There is not much difference between the cells. That means at a cellular level, we are not so different from parasites and the fungi. That means our cells are pretty much like them. Whereas the bacterial cells are relatively different because they are prokaryotes and we are eukaryotes. Okay. And what you need to understand why we are learning this is to make an antibiotic. If you have a difference, then that's better. We can make the antibiotic more easily. In the structure of the organism and the human, there should be some difference that is present. So who's generally different from us? As I just told you, bacteria. Bacteria are quite different from us. They are the prokaryotes. Okay. And if you talk about the fungi and the parasites, these guys are the eukaryotes. Is that understood? Now, try and understand this next part, what I'm telling you now. See, the point is this. When we talk about the differences, I hope you remember viruses are the only ones that either has DNA or RNA. That's why we classified the virus as DNA viruses and the RNA viruses. Okay? The remaining everybody has both DNA and RNA. Now, as I told you, if you want to make an antibiotic, there should be some difference that you should have. And who are different from us because we are eukaryotes? Bacteria. Bacteria are prokaryotes, so they're different from us. It's easy to make antibiotics on the bacteria topic. See, each thing from now will have one type of antibiotic. So if I get into this part, each of this, there would be one type of antibiotic. 1, 2, 3, and 4. In this topic, the antibiotics are present. Since DNA and RNA, they are called as nucleic acids, the antibiotics that are working here, they are called as nucleic acid inhibitors. Are you understanding? The antibiotics at this topic, they would be called as nucleic acid inhibitors. All right. Next, if you talk about the ribosomes perform protein synthesis, right? Ribosomes perform protein synthesis. So the drugs working here, they would be protein synthesis inhibitors. Next, just like that, you have a cell membrane inhibitor and cell wall inhibitor. So altogether, you get 4 types of antibiotics in bacteria, primarily nucleic acid inhibitor, protein synthesis inhibitor, cell membrane inhibitor, cell wall inhibitor. Because they are different from us, it's very easy to make antibiotics at them. Okay. Now anyway, let's talk about the ribosomes. Remember, I said that eukaryotic cell will have 40S and 60S ribosome, yes or no? And collectively, they are called as 80S ribosome. Please don't assume the 80S has been broken down into 2 pieces, 40 and 60. It's not like that. It's just a collective name given to them, 80S ribosome. Now, if you look here, this is nothing but 70S type of ribosome. So basically, bacteria have a different type of ribosomes compared to human. So it's easy to make antibiotics. And we have bacteria having these ribosomes, 30 and the 50. Bacterial ribosomes are 30 and the 50S ribosomes. I hope that's clear enough to all of you. Okay. Next, if you talk about fungal cell membrane, yesterday also we talked, it's made up of ergosterol. Fungal cell membrane, yesterday also we talked, it's made up of ergosterol. And in the parasites, it has cholesterol. So they have some kind of a sterol, right? Cell membranes are having some kind of a sterol, whereas bacteria don't have sterols. Bacteria don't have any sterol in their membrane. Last and the final, we have said that cell wall in the fungi is made out of chitin, and parasites do not have cell wall. No cell wall in parasites. So there are some differences between them as such, as you notice. Okay, that's what we talked about right now. Okay. Next, let's all continue our discussion from here. See, the point is this. Now we will talk about normal flora. That means usually in our body, there are some bacteria that are present, in or on our body. First one, on the skin, the bacteria that is present is Staph epidermidis. Look at the word, what is there? Epiderm. So it's very clearly telling you I'm present on the epiderm, I'm on the skin. So Staph epidermidis is that guy. Next is Staph aureus. Okay, next is Staph aureus, the other one that is present on the skin. Next, if you talk about vagina, in the vagina, the bacteria that is present is lactobacillus. Okay, the bacteria that is present in the vagina is lactobacillus, and lactobacillus is also known as Doderleins bacillus. Okay, it's also called as what? Doderleins bacilli or Doderleins bacillus. Clear enough to all of you? This is the one that actually maintains lactic acid inside the vagina. Lactobacillus produces lactic acid. Hence, the pH of vagina normally is acidic in nature. Is that clear enough what I said? That's why the pH of vagina, it is acidic in nature. That's how vagina is. So the point is, as long as this bacteria is present in the vagina, the pH would be acidic. If it dies, then acidity cannot be maintained, and now the pH becomes alkaline. Make sense? This guy maintains acidity. If it's not there, then the pH of vagina becomes alkaline. Is that understood? And that's where an infection comes by the name of bacterial vaginosis. We are going to learn about this later. Bacterial vaginosis comes when the pH of vagina becomes more alkaline. Okay. Now, any guesses who's the normal flora in the stomach, then in the CSF, then in the blood? Okay. See, the answer to this I'll give you right away. There is no normal flora in any of these parts. There is no normal flora in any of these areas. Are you understanding? There is no normal flora in stomach. There is no normal flora in CSF. There is no normal flora in the blood. I'm not denying the fact there can be some bacteria who can come. I'm saying normally they're not present. Normal flora are not present. That means if organisms comes in this area, definitely you're having some disease. That's the reason. CSF is 100% sterile. There should not be any organism in your CSF. If you're finding some organism, it means that there is somebody causing meningitis. So when there is meningitis, that organism would be coming in the CSF sample. Is that understood, doctors? So that's that. Stomach. A lot of people think either the E. coli is the answer, which is wrong. E. coli is in the intestine, not in the stomach. And H. pylori is also not the answer. H. pylori is not a normal bacteria in the stomach. If it is there, it's causing a disease called as peptic ulcer disease. That means even H. pylori is not a normal flora. Okay. So that's about that. As simple as that. Next. what are the things that goes on as such in microbiology for detection of organisms? See, there are a couple of things we do, right? One is culture. We grow the organisms on something called as culture media. And when we grow them on culture media, they may produce some color, some pattern of a colony, etc. So those are the ways we identify, oh, this must be the organism. We can do staining. So many stains are there. We can visualize them under the microscope, some motility, something like this can be seen. So these are the basic things: culture media, staining, and microscopy. These are the ways we can do the identification of this organisms. Are you understanding this thing? All right. Now we will start with one of the first things, that is the identification by some biochemical reaction. Okay. So first of all, imagine there are two bacteria. Bacteria number 1 has one enzyme catalase. Bacteria number 2 does not have the enzyme catalase. Okay. So two bacteria, one of it has catalase, one of it does not have catalase. So what is the job of catalase? Let's check it out. Catalase enzyme converts hydrogen peroxide to water. Are you understanding? It converts hydrogen peroxide to water. So that's the thing that you all got to understand. Catalase enzyme, it converts hydrogen peroxide to water. So is that understood? So now, out of these two bacteria, bacteria number 1, bacteria number 2, one of them has ability to convert H2O2 into water, hydrogen peroxide to water. So how can I know this? How can I know this? What do? Take a test tube. Take a test tube, put hydrogen peroxide into this. Put hydrogen peroxide into this. And now you put this organism into this. If the organism is having catalase enzyme, as I'm showing here, it would convert your H2O2 into water. That means you will start seeing bubbles in this area. If let's say organism does not have catalase enzyme, nothing will happen. H2O2 remains as H2O2. Okay, everybody understood? So all I'm saying is there are two types of bacteria. Some of them have catalase, some of them don't have catalase. How am I going to check for that? Take a test tube or a slide, anything you can take, add hydrogen peroxide to that. After adding hydrogen peroxide, add the organism who you are suspecting may have catalase. If it really has catalase enzyme, it will convert this thing, that is hydrogen peroxide, to water. All of you got this idea what I said just now? So please write this reaction. Catalase test. What is happening in the catalase test? Catalase enzyme if the bacteria has, okay, catalase positive organisms. Okay, so what will happen? Catalase positive organisms, because they have catalase enzyme, they can convert hydrogen peroxide to water. So catalase positive organisms, you will see bubbles. Bubbles would be seen. Catalase negative, no bubbles. That's a test by which I know, some organisms who have catalase, I can figure out, yeah, this must be the organism. Okay, so that's about that. Catalase positive bubbles would be produced. Catalase negative bubbles will not be produced. So these are the two things right here. Next thing, please understand this. This is the word that you got to memorize. The mnemonic is places, and you're going to just remember cat goes to places. Cat goes to places. So you remember and join the mnemonic. Cat goes to places means catalase positive enzyme. That means these are the organisms. In the entire micro, more or less, these are the important organisms who have catalase. So if I did this test and the bubbles came positive, so many organisms are there in micro, yet I'm just going to think only in this organism now. See, when we do the testing of organism, it's not one test that will tell me this is the organism. I have to do multiple tests in the lab, finally coming to the conclusion, oh, this must be the organism, step by step, step by step. So here we are. Who are the organisms that are catalase positive? At least put a star to 3 what I'm telling you. First one, Pseudomonas, Enterobacteriaceae, and then Staphylococcus. Okay, Staphylococcus. Pseudomonas, Enterobacteriaceae, and then Staphylococcus. So these are the three things as such. Okay, so 3. Cat goes to places. Cat goes to places. So Pseudomonas, Enterobacteriaceae, and Staphylococcus. 3 at least important guys you should know. So these will produce bubbles in simple language. Anybody who has catalase, all the places mnemonic organisms can produce bubbles. That's how I will know it's one of the places mnemonic. After I did this test, I'll do one more test. Then I will come to know, oh, this must be the exact organism. All right. So this is how the testing goes on. Next, write down this organisms called as catalase positive organisms, that is the mnemonic places. They usually cause infections in a disease called as CGD, and this CGD stands for chronic granulomatous disease. Okay, the word is chronic granulomatous disease, CGD. So in them, these organisms tend to cause the disease more commonly. Okay, so I hope that's understood by everybody. Done. Next, urease test. This is another biochemical reaction. Again, two bacteria, one having urease, one not having urease. So urease positive organisms, what they can do is, so simply write urease positive organisms, they can convert urea to ammonia, meaning they can create alkalinity for themselves. Whoever is having urease enzyme in their body, that organism, what can it do? It can convert the urea to ammonia. It can convert the urea to ammonia. Ammonia is what? Ammonia is alkaline. Is that understood? Ammonia is alkaline. So this is the word that you need to be thorough with. All right. So all these organisms can produce alkalinity for themselves. Now you just need to remember one thing. If the urease test is positive, pink color would be seen. If the urease test is negative, yellow color would be seen. See, actually we are starting with this only. The urea color is yellow. The urea color is yellow. If urease enzyme is present, then it is going to turn to which color? Pink color. If urease enzyme is not present, it will not be able to convert to pink color. So as simple as that, what we are saying is this: if that urease enzyme is present, the yellow color will be shifted to which color? Pink color. Yellow will be switched to the pink color. And that's our mnemonic if you want to remember the term or the word called as yep. Okay, so you're just going to remember this by putting this into the picture here. Yellow converts to pink. Yellow converts to pink if urease enzyme is present. So what is the whole idea here? Why are we doing all these things? We are doing them to actually do the testing. We want to identify organisms. So don't you think we need to know which organisms have urease enzyme who can, produce alkalinity for themselves? Let's go ahead. Okay, so these are the organisms that you need to know as such. There's a mnemonic again for that. But in my opinion, you should remember 3 guys what I'm telling you. This, and this. Okay, 3 guys that are basically least I'm saying. If you want to memorize, these are the guys who can create alkalinity: Staph, Protease, and H. pylori. And this is the explanation H. pylori, see, why was there nobody surviving in the stomach? Because of high acidity. Because of so much acidity, nobody is surviving in the stomach. Do you all agree? Then how come H. pylori is the only one who can survive? Because it has which enzyme? It has urease. It takes urea from the body, convert it into alkalinity. I just told you, what does these guys do? They convert it into ammonia. Urea is converted to ammonia, which is alkaline. So technically, in such a acidic environment, what is H. pylori doing for itself? It's creating alkaline media, neutralizing this thing. Hence, H. pylori can survive in your stomach. Clear enough to all? That's how H. pylori survives, because it has which enzyme? Urease enzyme. Hence, that's the reason for H. pylori diagnosis. I'm sure you have learned urea breath test or urease test. This is the testing you learn in internal medicine. Urea breath test or urease test. For which organism? It's for H. pylori. This is for H. pylori. Okay. So I really hope that this is clear enough with everybody. Okay. Now let's move on to the next thing. Stains. Why do we add stain to something? To increase the contrast. For example, right now this is white color as you're seeing. Okay, so if assume I add a color and that organism picks up the color, you can very clearly see against this background. So that is why we add stains. As simple as that. It will give a contrast to our organism. If they pick up our color, then that's what is going to happen. Okay, so there are different types of stains as such. We already saw India ink stain. Okay, India ink stain. All right, so what did we study in the India ink stain? You know, this is the stain for the capsule. This is the stain for the capsule. But what is special is when you add the stain, everybody else is becoming stained except for the capsule part. Are you understanding? When I added India ink stain, capsule is not staining. Everybody else is staining because everything became dark. The capsule is illuminated easily. Are you understanding this? India ink stain, it is a special stain. It comes under what stain? Negative stain. Negative stain is that stain in which the subject of interest. What is the subject of interest right now? Capsule. Capsule is not picking the stain. Everybody else is picking up that stain. So that's why what ends up happening is this capsule get illuminated. All right. So that's your India ink stain, the negative stain as such. Okay. Next. So technically, some structures do not easily pick up our routine stains. Some structures do not pick up our routine stains. All right. So for example, they're too thin to pick up any stain. So that's how they are very thin. They're not ready to pick up our stains. So only few stains have been reserved for them, which they can actually pick. For example, spirochetes. They're spiral. They're very, very thin. They don't pick up gram stain. Okay. Spirochetes, they don't pick up your gram stain. So we add a stain called as the silver stain. When you add silver, this guy will now actually become thicker. Now it becomes easily visualized. So these are some special stains again, as such what I told you. Okay. So there are two only, silver and Leveson-Ryu stains. But anyway, max to max, they can ask you silver fontana. So just keep in mind, because we cannot gram stain the spirochetes, we have to use what now? The silver stain, which can easily be infiltrated into this organisms. Okay, so that's that. Now we are coming to these things, that is gram stain and acid fast stain. See, this stain I'm anyway going to explain to you in diphtheria. This is a very special stain on which diphtheria shows this characteristic pattern called as Chinese letter pattern. So we're going to talk about this later. This is the special stain as such. Now let's write these things down, please. What are the steps that we need to do as such in gram stain and the acid fast stain? Okay, all of you first look here, please. These are the stains in which 4 steps need to be done, right? You can't stop in between. What is the purpose of the stain? This stain will identify whether the organism belongs to gram positive family or gram negative family. It doesn't tell you which bacteria it is, but it tells you which family is it from. Is it gram positive? Is it gram negative? See, when a sample comes, so many bacteria can actually cause the presentation. Okay, so what is our first step we do in the micro lab is we do gram stain. See, plain and simple, the moment the gram stain result comes, if it's gram positive, you have to think only in these many organisms. If it's gram negative, then you have to think in these many organisms. But if you don't do the gram stain, you don't know where to start. Okay, so many organisms are there. For example, if a sputum sample comes from pneumonia, so many organisms can cause pneumonia. How should I start? Where should I start? That's where I do gram stain. If result comes gram positive, I know in my head, these are the organisms who can cause pneumonia. Probably it's one of this. So I will do some special test for those organisms only. Are you understanding my point here? Okay, so how exactly do we do the gram stain? 4 steps. First of all, once a sample is collected, and if you put it under the microscope directly, they would be colorless. Organisms will not be able to be seen easily. They would be more or less colorless. Is that clear enough to all of you doctors? They would be more or less colorless. So that's one word that I wanted you to know. So that's why what do? We add stain now. First one, we will add a purple color. First color I will add is purple. So when I add purple color, no matter if it's gram positive or gram negative, both of them they will turn purple. And if your intention is to differentiate them, if you want to know if it's gram positive, gram negative, you can't stop here because both are appearing purple. We don't know. We don't know who's in the sample. When you add the purple color, if it's gram positive in your sample, that will also be purple. You know, the other one will also be purple. So next step what we do is, after adding the purple color, this is a procedure. You can't stop in any step. You have to do all the steps. We add a thing called as fixative. Fixative. Grams iodine is the fixative. Once you add the fixative, the color gets fixed only in the gram positive. Okay, the color will get fixed in which organisms? In the gram positives. In gram negative, color is not fixed. Just look at the star. Please remember, color got fixed in the gram positive. Now, when you add an agent, third step is to add an agent which will remove color, called as decolorizer. When you add decolorizer, one of them loses color. Which one lost the color? Gram negative. Why did it lose the color? If you look in the previous step, color was not fixed. But who retained the same color despite adding decolorizer? The gram positive, because in the previous step, the color was fixed. Are you understanding? Now, last step, we have to add, an opposite color. If you started with a purple, we add a pink. Okay. If your organism is gram negative, then what will happen? What will happen is, doctor, that organism will pick up which color? Pink color. Because that was the one who lost the color, now that guy will pick up the new color also. Okay, is that understood? Gram positives will retain the same color from the first step till the last step, and gram negatives will be regaining a new color. So, if you got what I said just now, pay attention. This part is the most important. At the end of the gram stain, what is the color of each of them? Gram positive will stay purple, or let's say it's purple only, and gram negative would be pink. Gram negative is pink, gram positive, it is going to be purple. Okay, so that's about it. That's your gram stain. Okay. Exactly opposite is acid fast stain. Exactly opposite would be the acid fast stain. What do I mean by opposite? See, in gram stain, you started with a purple or blue, then you ended with a pink. New stain was pink. Yes or no? You started with a purple, ended with a pink. But here you will start with the pink and you will end with the blue. Steps are same only. First one stain is added, then fixative, then decolorizer, then the opposite color. That's all. And that's where you will get the result. Acid fast organisms would be pink in color. They are positive, they are pink in color. And just like Gram stain, see, the positives called as acid fast, they retain the same color from the first step till the last step. Are you understanding? Always the negatives will lose the color and they will regain a new color. So who will regain a new color this time? Non-acid fast organism. Non-acid fast is blue. So see, you have to just spend 1 minute here. Main things to be remembered is this. Gram stain, the colors are this. Gram positives are purple, gram negatives are pink. Gram positives are purple, gram negatives are pink. And if you talk about this one, here what you need to understand, doctor, is this. Acid fast is pink, non-acid fast is blue. And finally, which staining is done more commonly? The answer is gram stain. Gram stain is very common, acid fast is rare. Why is it rare? There are very few organisms who are acid fast. Only if you suspect those organisms, then you should do acid fast. For example, TB. Mycobacterium TB, you must all know it is acid fast. So the point is, if somebody presents with a cough, chronic cough, blah, 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 with all the symptoms, then only the attending doctor will write a test, go get your acid fast stain done. Are you understanding my point here? You're not going to order the acid fast stain for every organism. You will only write it for those organisms which are suspected to be acid fast, which are very few, by the way. Gram stain is our everyday stain, right? Every day so many samples come. How to rule out the organisms? By doing the gram stain. So taking you back to this, if you understood all these things, let's go back, write all these things right here. See, this is the typical staining. Gram stain, we are staining peptidoglycan. Acid fast staining, we are staining mycolic acid. Not everybody has mycolic acid. Mycolic acid is present in very few organisms. Only they will become acid fast. You started with a purple here, yes or no? But here you will start with a pink stain. And what is the name of this pink stain? Carbal fusion. So this is a pink colored stain. Okay. So here you will start with carbal fusion. Now, fixative in gram stain, it's called as grams iodine. It's not difficult to remember, grams iodine. Here we have to heat the slide. Okay. In the acid fast, we have to heat the slide on which we are doing the testing. That's the fixative. Next, to remove the color, the agents are different. Here in gram stain, it is either alcohol or acetone, and here it is sulfuric acid. Okay, it would be sulfuric acid. And last and the final, the secondary stain. Here, the pink color is added by the name safranin or carbal fusion. Both are pink in color, whatever it is. You add either safranin or carbal fusion, both are pink. Here you will add blue. Once again, check it out. In the gram stain, you are starting with purple, you are ending with pink. That is what is the second stain. Second stain is the one we end on. That's the pink color. Whereas in acid fast, you start with the pink and you end with the blue. That's why the last stain is the blue color one. Okay, so that's your staining called as acid fast, and this thing what we call as the gram stain. Okay, now that you're all thorough with this, look at this picture, doctors. If you ever happen to see this cloudy background like this, it looks like it's about to rain. And against this background, when you see this pinkish organisms, that is acid fast. So cloudy, if you see, that is acid fast stain. You should recognize. Cloudy background is acid fast stain. And against this organism, pink organisms can be seen very, very clearly. Is that understood, doctors? The pink organisms can be seen very, very clearly. So that's about that you needed to know. Okay. So who are the organisms who are considered acid fast? There are very few organisms, as I told you. See, how should you start memorizing what I'm saying? Okay. So the words are this only. You may strike off this also, nothing matters. 3 mycobacteriums and 3 cysts. What are the 3 mycobacterium? Mycobacterium TB, atypical mycobacterium, and leprae. 3 mycobacteriums are acid fast and 3 cysts are acid fast. What are the cysts? Isospora, cyclospora, and cryptosporidium. Yesterday in parasito we learned this. I told you now, pink color cyst if you see, that is the cyst of 3 organisms who can cause diarrhea in HIV. Remember, cryptosporidium, isospora, cyclospora, they cause diarrhea in the HIV patients. So that's all. So you have TB, atypical, and the leprae, 3 mycobacterium and 3 cysts. That much only. Only these organisms in entire micro will appear which color? Pink color. They will be appearing in the pink. And please remember, it's not baby pink color. It's a deep pink color. It's a deep pink color. So that's what that you need to understand right here. All right. So that's about it. That's your acid fast stain and the gram stain. I'm just sparing a minute for you all. Please go over the gram stain and acid fast stain once quickly. See, make sure that you get the colors in the end. How to recognize. If I show you a sample, how will you recognize what I'm showing you? That's what I want you to know. Okay. Okay. So let's continue here, guys. Now what we're going to learn, our routine staining, that is gram staining, and definitely you have to know this for your exam, which organisms are gram positive, which organisms are gram negative. So what you have to do is, since there are too many bacteria, we'll just memorize the relatively smaller list. Once you're thorough with that small list, everybody else would be in the other list. So what is the story? Let's check it out. Gram positive organism, gram negative organism, which ones are we going to memorize? Answer is gram positive, they're less. Okay, and you must be knowing bacteria are going to be divided into 2 things, right? Based on their shapes. They're of 2 types. What are the 2 types? Cocci and the bacilli. Cocci, as you know, they are round. Okay, cocci means round. Bacilli means they are rod shaped. Gram positive means purple, gram negative means pink. Gram positive is purple and gram negative is pink. Is that understood what we said here? Okay. Now please understand one point what I'm saying. Gram positives, as I said, purple color and round. Purple and round, that tells me it is gram positive cocci. The moment you see that thing, you should start thinking either it could be a genus called as Staphylococcus, the name is coccus itself, right? Staphylococci, or Streptococci. Staphylococci or Streptococci. That's what you got to know, doctors, right? Staph and strep, these are the guys. The difference is one would be in cluster, one would be in chains. One would be in the cluster, one would be in the chain. So let me just show you that over here to you. See, look at this slide, what I'm showing you right now. Can you see that it's purple organisms, and what is the shape of the organism? They're round, and that too they're in cluster. The moment you see this kind of a slide, you should suspect most probably the organism is what? The organism is staph. If the same thing was in chains, then you should start suspecting what? It must be Streptococcus. So staph and then strep. Clear? Next, what you got to do is you need to sit back and memorize who are the gram positive rods. A, B, C, D, L, M, N. These guys are the gram positive rods. Okay, who are these gram positive rods? A, B, C, D, L, M, N. Clear enough to all of you? All right. Now, once you become thorough with this list, see, staph and strep are easy. You should know they are gram positive, right? That's very, very basic. And then the point is ABCD element. ABCD element. You have to know anybody apart from this. They all have to be gram negative. They all have to be gram negative. And now in the gram negative cocci, one organism you should write down. This guy would be pink now. This guy would be pink, and it would be rod, round like this. These are the gram negative cocci. These are the gram negative cocci. They're round and they're pink. If they're round and they're purple, what you just saw, that means they were these organisms on the other end. Is that clear enough to all of you? So just the color helps us differentiate whether it's this or this. Yeah. So that's your gram positive, gram negative organisms. Now, coming to this important thing, what we call as bacterial growth curve. Okay. So next thing in the list here is something by the name of bacterial growth curve. What do you have to understand here? What is bacterial growth curve? Let's see. Okay. Now the point is, bacteria can grow in the human body also. Bacteria can grow as such in this also. That is culture media. Okay. They can grow here also. They can grow in the culture media also. Okay, is that understood? Now we are going to just see the growth of these guys on the culture media. So we'll be starting that. And as usual, guys, just like I gave you time just to read the gram staining. Now I'm giving you 1 minute. Please read what is ABCD element and staph and strep. Once you memorize, we'll go forward. Be quick. Keep saying ABCD element. You read it like this: actinomyces, bacillus, clostridium, diphtheria. Like that, you have to go memorize that in a minute. Give it a reading at least for now. Okay, give it a reading, and we will begin shortly. Okay, so I hope you guys are done. Now let's start bacterial growth curve on culture media, right? See, we grow them artificially by providing them food outside the body. Now we can grow them outside also. See, why are they coming into the human body? They get their nutrition. If you leave your food somewhere, some fungus, somebody grows on that. That's nothing but culture. Okay, if you happen to leave a bread piece somewhere, after some time you will see growth of fungi. You leave some curry somewhere, you will see growth of some organism on that. So that's what is the culture, right? Directly or indirectly, you guys have all performed the cultures at one point of time in your life, yes or no? Right? So when you are lazy, that means you have performed the culture. Now here we are. In the lab, we do it intentionally because we need to do the diagnosis. So that time we have to provide them with the foods that they like, and we grow the respective organisms. Okay. Now, what is the story is, let's say I collected a sample from somebody. The sample will not have as many bacteria, it will have little bit. So now the point is, those sample, I will take that sample little bit and put it on my culture media. If bacteria are present in that, they will start multiplying. Yes or no? They will start multiplying on your culture media. So that's what we are going to notice. What is the pattern of their growth on the culture media? Starting from here, doctors, when you put them on culture media, they don't start reproducing right away. That's why the line is going to appear flat like this, right? They're not reproducing. That's why the line appear flat. This is called as lag phase. A lot of times when your phone is stuck, what do we say? My phone is lagging. It means no activity. It's not doing anything. It's lagging. Lag means no activity. Bacteria are not reproducing right away. But what are they doing is, since they have seen so much food is abundantly available for them, they are now getting ready to reproduce. They're not reproducing right now, but they're getting ready to reproduce. To get ready to reproduce, what do they have to do? They need to increase the enzymes of reproduction. So that means in the body, they're accumulating the enzymes. Because of this, the size of the bacteria become bigger. So maximum size of the bacteria, it will be seen in the lag phase. The maximum size of the bacteria is noticeable in what? It's noticeable in the lag phase. Next, what happens once they start reproduction? Now they go crazy. The number of bacteria start going up and up exactly like this. The number of bacteria start going up. And please understand, this is known as log phase. This is known as the log phase. Now in the log phase, dear friends, what you need to understand is there is massive reproduction that is happening. Because of continuous reproduction, obviously all the enzymes in the bacteria are used up now. They accumulated the enzymes for reproduction, but now all those enzymes have been used up. That means now the bacterial size becomes small. The size decreases. Okay. And assume this is the culture media and I want to do the staining. I can take some sample from the culture media and I view it under microscope. This is the best place if you want to collect a sample. Okay. Log phase, because they're all easy to simply collect and visualize under the microscope. Next, third phase. The third phase is what we call as what? Stationary phase. The third phase is called by the name of stationary phase. Is that clear enough what I'm saying? Third phase is called by the name of stationary phase. Now what is the story about the stationary phase, doctors? See, stationary phase is that phase where births equals deaths. Bacteria are born also, but they're dying also. Why are they starting to die now? Because too many bacteria have been produced and the food is less now. Food is scarce. That's the reason the bacteria starts to die. So now births equals deaths. Births equals deaths. This is known as stationary phase. And do you remember, yes, today when we talked about this, we said whenever the conditions are bad for the bacteria, the organism form converts to the spore form. The organism forms converts to the spore form. So this is the word you need to know. Stationary form. Stationary stage is the one in which what is happening? Spore formation is taking place. And then finally, you have something by the name of death phase. So finally in the story, what do we have is, we have this thing by the name of what? Death phase. Death phase means eventually all the food is used up and now slowly all the bacteria start to die. The way they were reproduced and now they will start to all die. So that's the story that you need to know as such about them. Clear enough? So 4 phases, how the questions can come. This is an important topic I already has marked for you for question. They can ask you arrange the following bacterial growth curve in sequence. Lag, log, stationary, death. This is one thing. Second, they can ask you size of the bacteria is maximum in answer lag phase because they are accumulating the enzymes of reproduction. Second, where is the size less? Log phase because they have already used up those enzymes. When is the bad phase for the bacteria? Stationary phase. That's where they will start to form what? They will start to form the spores. Okay, that's that. Next and the final. This is about the bacterial reproduction. Check it out. Bacteria, they reproduce by this process called as binary fission. Bacteria, they reproduce by this process by the name of what? Binary fission. Okay. Now, what is binary fission? Each time they reproduce, they double into their numbers. That means 4 bacteria becomes 8, 8 bacteria becomes 16, 16 becomes 32. Like that, each time they're reproducing, they're becoming double their number. So, what is their doubling process called as? Binary. Bi means 2. Each time they will become twice their numbers. Okay. That's the way bacterial reproduction happens. Now, don't you think for them to double, some time would be needed here? Some time would be needed. This time is called as what? Generation time. How much time they need to reproduce? What is this called as? Generation time. Right? This is called by the name of generation time. Okay. How much time do most bacteria take to double up? 20 minutes. 20 minutes. Every 20 minutes they are doubling up. That means within 1 hour only, they have reproduced 3 times. They have become double, double, double 3 times. Are you understanding? So that's why, if suppose from a patient I collected a sample, some sample, I don't care. That sample I put it on a culture media. How long does it take for me to see a visible growth of those bacteria on the culture media? See, first of all, this is the visible growth. Are you seeing the bacterial colonies right now? They can show some color, they can show some pattern. Based on these things, we can identify which bacteria it is. So now, how long will a culture report take place? The answer is 2 to 3 days. The culture reports usually come back in how much time? 2 to 3 days. So this is something that you all need to get familiar with. Okay. And now, bacteria, based on a couple of things like oxygen requirement or temperature. Based on oxygen requirement or the temperature, they are divided into these three types, as you can see. Okay. So that's about that. See, it's not very high yield as such. We need to just get an idea. Some bacteria, they completely want oxygen, otherwise they will die. Obligate means compulsory. Obligation means compulsion. Without oxygen, they won't survive. Obligate anaerobes means they don't like oxygen. If you give them oxygen, they will die. Microaerophilic means somewhere in between. They're not aerobes, they're not anaerobes, they're somewhere in between. They're called as microaerophilic. Are you understanding this point here? Okay, same way, bacteria don't like extreme temperature. Bacteria like which kind of temperature? Somewhere in between. And middle layer is generally called as meso. Ectoderm, endoderm, mesoderm. Meso means somewhere in middle. They don't like very high temperature also, they don't like very less temperature also. They like the moderation of temperature. Are you understanding? So, which is the most comfortable state for bacteria temperature point of view? Mesophilic state. And then if you talk about organisms in general, most bacteria do not come in either of these categories. Are you understanding? Most bacteria do not come under this. These are special guys. So what do most bacteria want? Answer is they don't want anything. Most bacteria, you can write the word, most bacteria are facultative. Okay, most bacteria are facultative. Is that understood by all of you? Most of the bacteria are facultative. That means no special requirement. They will survive in oxygen also. They will survive without oxygen also, with less, whatever you give them. They don't complain. They survive easily. And most of the bacteria are like that. That's why they can sustain in the environment more easily. These are the people who are very finicky. I want this only. I want that only. These people will not survive easily. Are you understanding? So this is all it is. So you should know who are these anaerobic organisms. Just write these three names, please. Actinomyces, Bacteroides, and Clostridium. Actinomyces, Bacteroides, and Clostridium. These are the three guys that are actually the anaerobes. They are the anaerobes. And if you had memorized the list I gave, ABCD element, this guy is gram positive, this guy is gram positive, the only gram negative is Bacteroides. So there are 2 gram positive anaerobes and there is 1 gram negative anaerobe, and that is Bacteroides. Okay, this one you leave it for now, later I will tell you what it is. Alright friends, so okay, we will call it a day for today now. Alright, we have around 15-20 minutes remaining. I think the class was informed till 4, right? So we will stop here for today, right? Thank you very much. I will again see you all tomorrow.